Hi guys, who's ready to talk about enzymes? You are, and I am. All right, so remember that enzymes are proteins, and remember that it could either be that they're at their tertiary structure, or if there are multiple polypeptides involved together, that could be their quaternary structure. So if we just have one polypeptide that has gotten itself all coiled up, it's at its tertiary structure, but it might be multiple polypeptides, in which case we're at our quaternary structure. And what enzymes do is they act as a catalyst, so they speed up chemical reactions. And another important thing to remember is they're not consumed, they're not used up, they're totally reusable. And then we need to remember that they are not permanently changed. I'm going to talk to you about a little bit of a change, but they are not permanently changed. So there's a small change, but not a huge change. So proteins, remember, are these big, globby, messy things once they get all coiled up. And they just kind of look like this, like kind of a mess. So remember we said they're catalysts, and so they work like any other catalyst. What they do is they reduce the activation energy that's necessary for whatever chemical reaction is going to take place. Because remember, every chemical reaction has to have some kind of energy to get it started, and that's that activation energy. So if we take a look at this graph, we can see what it looks like. So this black line shows us if we didn't have an enzyme, how much energy it would take to get us started until then we get this downhill roller coaster and we get this huge change in energy. If we have an enzyme or a catalyst present, we only have to get to here for our activation energy, but we do ultimately still end up at the same place. We still start in the same place, we still end in the same place, we just don't need as much activation energy. That's what an enzyme does. What makes an enzyme different from any other catalyst is that an enzyme is a protein. It's a biological catalyst. So a lot of the catalysts you've worked with in the past have been things like zinc, which is just a metal. It's not biological. It's not a carbon-based biomolecule that's doing this. An enzyme is a carbon-based biomolecule, and it's a protein specifically. So the thing that's most important about an enzyme is the thing that's most important about any protein we talk about ever, and that is its shape. So as soon as you hear enzyme, your next thought is protein, and as soon as you hear protein, you know your next thought is shape. So enzyme, protein, shape, that's what's most important. And the reason it's important is because there's a binding site, and that binding site or that active site is where a substrate is going to come in and bind. So the substrate is whatever the target molecule is. So here's my enzyme, here's my substrate. The enzyme has to have a site that's the same shape as the substrate. If I have a different shape, the substrate doesn't fit and it can't do the reaction. So what can happen though is that this comes in, and when it comes to the enzyme sort of gives it a hug, and that's called an induced fit. And actually regular hugging works that way. So if you're standing like this and somebody comes up like this to give you a hug, you induce your fit and you put your arms around them to give them a hug. You don't just stand there like a big weirdo. I mean, you might, depending on who it is, but you probably don't just stand there. You probably are maybe coming in for the hug, and then you just squeeze a little tighter when you've got the person there. That's what an enzyme does. When that substrate leaves, the chemical reaction takes place, that substrate leaves, the enzyme relaxes a little bit, goes back to that normal binding site again. So here we have an enzyme. It's this big green thing. There's the active site that is this little kind of outside of the S outline. You have your substrate, which is conveniently shaped like an S in this diagram. And this could drag in here and fit right here, and the enzyme would hug around it and grab onto it and do whatever needs to be done to that substrate. So here's another example. This particular example, the enzyme is sucrase. Anytime you see ACE, you know you have an enzyme. So just like when you see OS, you know you have a sugar, ACE tells you you have an enzyme. So anytime you see ACE, you know you've got an enzyme, okay? So we've got our active site right here, and conveniently enough, when you see what sucrose looks like, so sucrose comes in and binds with sucrase, and notice that the shape of sucrose is the same as the active site in the sucrase. So sucrose is the substrate, sucrase is the enzyme, the substrate bonds with the enzyme gives a little hug, and when it gives it that little hug, we convert the products. So in this case, water comes in, gets added to break this bond. Take a second, hit pause, figure out what that kind of reaction is called. Go, pause. Did you really pause? Did you really think about it? Okay, it's hydrolysis. So remember hydrolysis, hydro, we add water, lysis to break something apart. So this particular enzyme is catalyzing a hydrolysis reaction. So we're going to add water in, and what we're going to see happen is that these products are going to get released, and we've got glucose and fructose because that's what sucrose is made up of. And then most importantly, we have our enzyme sucrase, which is not at all changed, not at all used up. It's ready to go again. So more sucrose can come in, more sucrose can bind, and when that sucrose comes in and binds, can get broken down again. Now, enzymes can break things down. Enzymes can also put things together. So it can go one of two ways. It can be a putting together, or it can be pulling apart. It just depends on what reaction it is that enzyme catalyzes. Every reaction has its own enzyme. Every reaction has its own enzymes. They're very specific. So every enzyme, there's only one thing it can do. Every enzyme only has one thing it can do. This enzyme, sucrase, can break down sucrose. That's all it can do. It's the only thing it can do. That's it. That's its only job. Okay, so what affects enzyme activity? Well, one of those sort of big categories is environmental conditions, and another one is the presence or absence of these things called cofactors or coenzymes. Now, for optimum activity, enzymes require certain environmental conditions. I don't need to tell you what they are, you already know, because as soon as I say enzyme, you think protein. And when you think protein, you think shape. And you already know what the three things are that can affect the shape of a protein. I'm going to tell you anyway, though, because I'm nice. But maybe this is a good chance for you to stop and quiz yourself. Do you know the three things that affect the shape of a protein? If you know the three things that affect the shape of a protein, then you know what the three things are that are going to impact the enzyme's activity. So hit pause, quiz yourself, go. All right, here we go. Temperature. Temperature is really, really important. If we're talking about human enzymes are going to function best at 37 degrees Celsius because that's human body temperature. If we're talking about some other organism, though, then 37 degrees isn't necessarily going to be the best temperature. Not all enzymes are the best at 37 degrees Celsius, just the ones that are in the human body. So every enzyme has its own temperature range where it's going to be effective and work well. If we're talking about humans and we get to a really high temperature, that's going to denature your human enzymes. So think about that for a minute. Think about a time when you might have a really high temperature, like if you have a fever. If you've got a fever and your fever gets too high, you're going to start to denature your own enzymes. And then your own enzymes aren't going to be able to take care of your own metabolic processes, and then you're in trouble. That's why, especially with babies, when a fever gets super duper high, we have to make sure we do whatever we can to lower that fever and take care of whatever it is that's going on, because that high temperature can actually cause all kinds of other problems. Most enzymes also require a certain pH. A lot of times around neutral. Not always, though. Pepsin's an example I'm going to show you on the next slide that does not require a neutral pH. In fact, a neutral pH is terrible for pepsin. And then ion concentration. So typically we're talking about our salt ions and our salt concentrations. Again, it just depends on the enzyme what kind of salt environment it needs, how much salt concentration it needs versus doesn't need in order to work well. And so every one of these enzymes has its own ideal range of conditions in each of these three categories. And if you go outside of that ideal range, you're going to have less enzyme activity. So pepsin is a digestive enzyme. It breaks down proteins in your stomach. So its job is to break down proteins. Pepsinogen is inactive, and pepsinogen is what's already there. Pepsinogen is 44 amino acids longer than pepsin. It's released by your stomach cells, and it gets activated by hydrochloric acid. So what happens is the hydrochloric acid comes in to the pepsinogen. So you've got pepsinogen, your stomach cells are like pepsinogen, pepsinogen, pepsinogen. And then the hydrochloric acid comes in, hydrochloric acid, hydrochloric acid, hydrochloric acid. That comes in, causes the pepsinogen to denature. When the pepsinogen denatures, it ends up in the right shape to be pepsin, and then pepsin can start to digest proteins for you. So HCl in your stomach is actually not there to digest anything. The HCl is not there to break things down. The HCl is there to create an acidic environment so that pepsinogen can become pepsin, and then pepsin can do its job. If you don't have the right pH, pepsin isn't formed, pepsin can't do its job, it's not the right shape, can't do what it needs to do because we've got a lock and a key, and the lock is not the right shape for the key. So the proteins come in and they can't fit into that pepsin lock in order to get broken down. Isn't that cool? All right, so there are also enzyme helpers. Some of them are called cofactors. Cofactors are inorganic. So those are things like zinc and iron and copper. So iron is a necessary cofactor for hemoglobin. So you might remember that when we looked at the hemoglobin molecule a couple PowerPoints ago, there were little heme groups and it showed that there was iron in there. Iron was a cofactor helping hemoglobin in order to hold on to the oxygen in your blood. Zinc, iron, copper, you see those things on like cereal boxes when they talk about how nutritious the cereal is, and it's because you need those in your body. So as weird as it is to think that you need zinc, you're not going to take a big handful of zinc pellets and chew them down, but you do need them to be in your food. Coenzymes are organic molecules. So cofactors are inorganic, coenzymes are organic, and typically we're talking about vitamins. So what happens is that you have your enzyme here. So here's your enzyme starting out. And then you have a coenzyme that comes in and it's going to fit really nicely right here. So once you have your enzyme and your coenzyme, the substrate can fit. And the coenzyme helps make that fit exactly the right fit with the enzyme itself. So the cofactor or the coenzyme just kind of improves the fit for the substrate so that then the substrate can come in and whatever reaction it is that needs to happen happens. There are also inhibitors. So if you think about what inhibit means to get in the way of something or to stop something from happening. So an enzyme inhibitor can keep an enzyme from working. Now, the enzyme inhibitors are actual molecules. So when we talk about those different environmental conditions, that's not an inhibitor. That's an environmental condition. An inhibitor is some other kind of molecule that's going to come in and do something. So here we have our enzyme, and here we have our substrate. And normally the substrate is going to come in and it's going to bind and it's going to fit nicely into this active site. But what might happen is somebody else who's kind of shaped like the original substrate can come in and actually bind in that location. And that's called competitive inhibition because that other molecule competes for the active site. So that guy just kind of swoops in and goes, nope, I'm going to sit here. You can't sit here. Like when you want the really good seat to watch TV and your sister goes and sits down in that seat that you really wanted and like you sit on top of her and then you guys start to fight, that's competitive inhibition. She's inhibiting you from sitting in the seat that you want to sit in. Okay. There's also non-competitive inhibition, and this one's kind of cool. So you notice that there's this other site over here. So with non-competitive inhibition, somebody can come in and sit into this other site over here. And when it sits into this other site over here, it changes the shape of the active site just enough that the substrate can't fit in there anymore. So when they come in, when this guy comes in and hits this other part, not the active site actually changes enough that the substrate doesn't fit. And if the substrate can't fit, it can't get in there and the reaction can't take place. So again, inhibitors are molecules, and they're molecules that are going to come in and either mimic the molecule itself, the substrate itself, and go into the active site, or they're going to go into some other part of the enzyme that's not the active site and change the active site shape enough that the substrate can't get in there. So here's an example. Methanol is an alcohol found in paint thinner. It's found in copy machine fluid. It's found in varnish. It's found in windshield wiper fluid. And it's something, interestingly, once ethanol was made illegal during Prohibition, people started to go to methanol. And they were like, I'm going to start drinking this. Great idea. Not such a great idea. So there are enzymes, including one called alcohol dehydrogenase, and that's what this one is. This one's alcohol dehydrogenase. You can totally recognize that, right? What they do is they convert methanol to formaldehyde and formic acid. Well, if you've ever heard of formaldehyde, that's what we used to preserve animals in. It's also pretty cancer-causing in huge doses, not the kind that's in your body when you're breaking down methanol. And formic acid is also gross, and we don't want those. And so we're going to get these other toxic things. And if you have enough of them and you can't clear it, you can end up with methanol poisoning. And the symptoms of that are blindness and breathing difficulty, convulsions, seizures, vomiting, all kinds of gross stuff. Okay. So if you've got methanol in your body, you want to make sure that you aren't breaking that down, because if your body gets a chance to break down that methanol, you're going to have this other gross stuff in your body too. And that gross stuff is going to poison you because your body is breaking it down. So what do? Oh my God, somebody just drank methanol. What do? Interestingly enough, what you should do is give them ethanol, which is regular drinking alcohol. Now, really, they don't do that anymore. There's another thing that they use that's kind of like that you would get if you went to the hospital because ethanol is too unpredictable in a body because of people's tolerances and stuff. But in a pinch, you give them ethanol. And the reason for that is that ethanol is a competitive inhibitor. So ethanol actually binds better with alcohol dehydrogenase than methanol does. So if alcohol dehydrogenase is sitting there and there's ethanol and there's methanol, it's going to grab the ethanol. And then when the ethanol gets broken down, it's not broken down into the gross things that methanol is broken down into. You don't get those poisonous things. You get other harmless things that then your body can clear and get rid of. Interestingly enough, ethanol is also a competitive inhibitor for antifreeze, which is ethylene glycol. So ethylene glycol is something that people sometimes, little kids sometimes will get poisoning because antifreeze, when it's in the garage, smells really sweet and it's blue, and so it looks like Gatorade, and so little kids will drink it. And again, I mean, you're not going to get them drunk, but ethanol is the antidote to ethylene glycol poisoning, which is kind of weird, kind of crazy. Like, here, have a drink and we'll take care of your poisoning. But it's all because of that competitive inhibition. And if you look at those molecules are pretty similar. And so you can imagine how they would share a similar binding site. Okay, so I am going to finish off my part of this now, but don't leave because I have another quick video that's going to take you through some animation and show you some cool stuff about how this works just a little bit better than I can with all of my little static diagrams. So hang out, watch the video, and learn some stuff. See you later, alligators.
Enzymes are protein molecules that speed up chemical reactions in the cell. A special region on the enzyme, called the active site, is the area where enzyme activity takes place. An enzyme works by binding to molecules, called substrates, at its active site. The job of the enzyme is to convert the substrate into a different product or products through a series of chemical reactions. Following the reactions, the products are released and the enzyme is free to act on another substrate. At the active site, the substrate should fit into the enzyme almost like a key fits into a lock. Because of this, most enzymes can fit only one substrate. To accomplish this lock and key fit, the active site undergoes a slight change in shape in order to better accommodate the substrate. This is called the induced fit model because the enzyme is induced to undergo small changes so that the substrate can achieve optimum fit. All enzymes have optimal environmental conditions that are correlated with maximum enzyme activity. Environmental conditions, such as pH and temperature, play a role in how efficient the enzyme is in conducting its reactions. When conditions are less than optimal, an enzyme will lose its configuration and slow its activity. This is due to a change in the three-dimensional shape of the enzyme and is called denaturation.
Oh my gosh, you guys, I can't believe I forgot to say a thing. I was just watching that video, that little animation thing go on, and I went, I can't believe I forgot to say a thing, and this is so, so, so important. And it pertains to this graph. I mean, only sort of kind of, I just didn't want my face to be like gigantormous on the screen, so I had to pick a slide for this to go with. But at any rate, the thing I want you to notice with this is that that black line shows you that this is a reaction that could take place on its own without the catalyst. So this could take place without the enzyme. So enzymes will only speed up reactions. They will not make reactions happen where they would not happen on their own ordinarily. So enzymes really just speed up reactions. They make them happen faster. They don't make them happen. So a reaction that would not happen on its own is not going to happen with an enzyme. The enzyme just makes it happen faster. Now, it might just be that because of those enzymes, our bodies can keep up with what they need to keep up with. And without those enzymes, our bodies can't keep up with what they need to keep up with. So that's the case. But these enzymes are only doing a job that could already be done. So they're only helping a reaction along when that reaction could already be taking place. They're not going to make a reaction happen when it wouldn't happen ordinarily. That's super important to remember. Okay, now we're done.